Good afternoon, and welcome to today's installment of the Wednesday afternoon lecture series. My name is Eric Billings, and I co-chair the Systems Biology Interest Group, which has the privilege of hosting today's guest speaker, Dr. James Collins. Dr. Collins received his undergraduate degree in physics from the College of the Holy Cross in Massachusetts and a doctorate in medical engineering from the University of Oxford. In 1990, he joined the faculty of Boston University, where he is currently a professor of biomedical engineering and co-chair of the Center for Biodynamics. Dr. Collins' work on complex systems has been recognized with the NIH Director's Pioneer Award, a MacArthur Fellowship, and he was recently selected as a Howard Hughes Medical Institute investigator. The title of Dr. Collins' talk today is Engineering Gene Networks, Integrating Synthetic Biology and Systems Biology. Dr. Collins.
Okay, well, I'd like to thank Eric for the introduction and the invitation to speak to you today. What I want to do over the next 45 to 50 minutes or so is give you a feel for some of the things our lab's been doing in synthetic biology and systems biology, really from an engineering perspective and now increasingly from a biological perspective. What Eric didn't know was my background's originally in physics, and I moved into medical engineering in grad school. In grad school, I actually focused on whole body dynamics. So we had initially studied things like human locomotion. When I first joined Boston University, we actually focused on human balance control, both coming up with physics-oriented techniques to characterize how people maintain balance, but also coming up with methods based on noise, so mechanical, vibrational, electrical noise, as a means to improve balance. So in these efforts, using concepts such as stochastic resonance, we're able to show that if you deliver subsensory mechanical noise to the soles of subjects' feet via vibrating insoles, you could substantially improve their ability to maintain balance. So much so we could take a 75-year-old and have them balance as well as a 20-year-old. And the significance for that, and we actually had NIH support, this was our prime focus in my initial R01, was on human balance control, is that, as many of you can appreciate, falling is a major problem for older people. About 1 out of 3 people fall each year, and with about 15 to 20% of such falls resulting in serious injury. So I had lunch with the postdocs as part of the program here, which was great, and they were asking me about my career trajectory, how did I choose what to work on, how did I get to where I was, and wanted to share with them this notion of, okay, how do you go from studying human balance control, which is effectively a dry lab notion, to looking at balance, kind of balance now at the level of genes and proteins. And it goes to basically the mid-90s when Charles Cantor, who I know is familiar to many of you in the room, was chair of our department. So when I first joined Boston University, I was on the research faculty, which had the downside that I didn't teach any classes, but had the upside I never had to go to a faculty meeting. Well, at one of these faculty meetings, Charles and the other faculty had decided that they wanted to go for a large development grant from the Whitaker Foundation. And the Whitaker Foundation was putting out multi-million-dollar grants that would initiate and develop new training programs for bioengineers. And Charles and the faculty decided they wanted to pitch one that would train bioengineering students to go from genes to cells to tissues to organs to whole body. So they made the pitch, and it was strong enough that they were invited for a site visit. So the site visit was coming, and the Whitaker Foundation, like many, including the NIH, liked to see early efforts in what was being pitched. Charles called me in his office and said, Jim, we're going to be embarrassed. We don't have anybody bridging these levels, but I know you're doing nonlinear control theory applied to the human body for balance control. Would you be willing to try that down at the level of genes and proteins and cell molecular biology? I said, sure. He said, great. I said, I got a good system for you. We've engineered a cell suicide system that's set up to gobble up something in the environment and then kill itself after the substance is gone. But it's leaky, and as a result, it's killing itself sooner than it should, and could you use your modeling to basically make predictions as to how could we improve this? I said, sure. So he went to his whiteboard and he started writing all these rate equations down, and I took them down. And as I'm leaving, he's all excited. So, Jim, I forgot to tell you something. You have 4 days to come up with your presentation. The site visit's on Friday. So I went running back to my lab, and Carson Chow, who's a PI here at the NIH, was a postdoc in my group at the time. And as I said, we were working on human balance controls. We didn't know any molecular cell biology. So with Carson and a few of our grad students, we began playing around. And 2 things to know. One was the equations had no interesting dynamics. So for the modelers in the audience, these were just fixed points. There really wasn't anything that was exciting. And 2 is it really had almost nothing to do with the biological system that Charles wanted to model. We didn't know enough molecular biology to make tweaks on it, but we tried to do what we could. I went back to Charles Thursday night. I said, Charles, I'm sorry, we're going to have to punt. We gave it our best shot, but we came up with nothing. And he says, no. He says, I want you to go out there tomorrow, Jim, and make your presentation nonetheless and tell them of your vision as to what an engineer could do for molecular biology. So I said, okay. So they wheeled me out the next day, and I gave them my 96-hour half-baked notions about what an engineer who didn't know any molecular biology could do in molecular biology. And we didn't get that grant. It actually became a fairly famous site visit, thanks in part to my presentation. Luckily, the department didn't hold it against me, and what happened subsequently, along with Carson and others in our group, is we began to think about what could we do as engineers. So this is about 96, 97, and at the time, we were strongly encouraged by a number of groups and a number of our colleagues to think about reverse engineering natural networks and signaling pathways. But if you think back, this was just as arrays were coming online. They were very expensive, and there was very little data that were available. And so the notion of inferring or reverse engineering a network from very little data was crazy to us. So we ran as fast as possible as we could away from that problem. But as bioengineers, as we had more than 96 hours to think about things, and as we began to read some of the textbooks and look at the papers, we thought instead of reverse engineering natural networks, as you would on inference, could we as bioengineers instead forward engineer synthetic networks? So could we take biomolecular components, whether they be promoters or genes or proteins or other bits of DNA, RNA, could we, as an electrical engineer, would take these components and put them together into circuits that were designed in the way that we wanted them to be designed, so they would be basically synthetic circuits. So the schematic would be kind of in the front end, similar to what you would do in an electrical engineering department, where you would come up with a schematic idea of a circuit of some interest. You would model it mathematically. Then on the base of that circuit, actually try to encode the network into a plasmid or into a construct, get into a cell, and see then if you could actually get it to work in the way that you designed it. So with Tim Gardner, who is a mechanical engineering undergraduate who joined my lab as a grad student, he and I began to think about interesting problems you might go after. And we considered a number of different networks and circuits, and then came up with one that was inspired by the notion of a toggle switch or flip-flop in electrical engineering. These are simple memory elements that are at the heart of your digital computers. And so with Tim and myself, we said, okay, could we make this a genetic version, so one that would be, say, simple enough that you could actually build and get it to work, but complicated enough that would have interesting dynamics that would be worthy of study. And so we came up with this notion that is shown schematically here, where you basically have two genes. You also have a third that's a reporter, but basically repressor 1, repressor 2. You set it up so that your promoters are constitutive. Okay, so it's designed so that each gene wants to be on. You then set it up so that the protein produced by gene 1 wants to bind to the promoter for gene 2, shutting it down. And the promoter produced by gene 2 wants to bind the promoter for gene 1, shutting it down. Okay? So in this system, you have both genes want to be on, but they're each trying to shut each other off. In principle, you can set this up if you have appropriately balanced genes or promoter strains so that you can have a bistable system. So that is a system that wants to exist in one of two stable states. Either state 1, where gene 1 is on, gene 2 is off, or state 2, gene 2 is on, gene 1 is off. Okay? In principle, you should then be able to flip it between the stable states by transiently delivering an inducer for the currently active gene. Okay? So let's say you're in state 1, gene 1 is on, gene 2 is off. What you should be able to do is deliver, say, a chemical inducer for that gene to now shut off its protein or stop being produced. This now allows gene 2, which had wanted to be on but was cut off by gene 1, to now begin to come on, produce its protein, and once its protein's on at a high enough concentration to now keep 1 off, you can remove your inducer, and you flip from state 1 to now state 2, where now gene 2 is on, gene 1 is off. Well, very nice in the abstract, and what we did was where we usually go in biomedical engineering, in our lab at least, is we turned to modeling. So we came up with some very simple dynamical models to consider, where what you have here is U and V are the concentrations of the proteins of gene 1 and gene 2 respectively. These are differential equations, so these are derivatives. Alpha 1 and alpha 2 are effective synthesis rates. And then you have measures of cooperativity of repression with beta and gamma. Okay? You can do some very simple dynamical analyses on these, of type you might, as a senior undergraduate, get in a math department or an engineering department or a physics department, where you would set these equal to 0, so you can look at steady states. You'll now have your null-Klein equations, where you can now plot in your protein UV space that, provided you have some nonlinearities, beta and gamma, okay, and if you've got basically that they're equally balanced or close to equally balanced, your sigmoidal plots in your UV space will basically intersect at 3 different points. corresponding to 3 different steady states. The first one is at separation. This is an unstable point, which is kind of that they'd be equally balanced. The system doesn't want to sit there. And the others are stable fixed points or steady states, where this corresponds to state 1, gene 1 is on, gene 2 is off, and this corresponds to state 2, gene 2 is on, gene 1 is off. You can do additional analyses, so-called bifurcation analyses, where you can, for example, look at the effective parameters. You can look in the plots of log-log of the effective synthesis rates to find regions for bistability versus monostability. What you find from very simple analyses is that these regions of separation between the 2 states are basically directly related to the slopes directly related to beta and gamma, indicating you have to have some nonlinearity in the system as a result of multimerization of your protein or cooperativity repression to get bistability. And that's captured nicely here. As you increase the nonlinearity, you go from a very small kind of cheese wedge region to a very large region. So if you're a grad student with, say, a 4-year NIH fellowship, you don't want to be in the lab exploring this region. Good luck. You want to be in this region to try to find it. Okay? Well, in the math world, biology or physics, you can give a whole talk on this, and actually people are quite interested. Appreciate when you come to NIH, as much as there's a big effort for computation, I appreciate within the NIH, both on this campus and broadly in the extramural program. At about this stage, basically people are looking, saying, who invited this guy to give this seminar? We're not mathies. You know, the docs in the audience will be faking, acting as if they've got a beep. Oh, I got to go. I got a patient that needs to see me. And Tim Gardner and myself realized that that would be the case. And if we wanted to have an impact, we would need to get past this and actually do experiments. And we had no background in molecular biology. Tim Gardner, as I said, was an aerospace mechanical engineer at Princeton. He designed autonomous helicopters as a senior project that he flew around the campus as his academic project. I had come from looking at human balance control. Well, what we did was we took our transparencies and found 10 of our friends who worked in molecular cell biology and basically asked them, presented what I showed you, what do you think? And invariably the discussion would go as follows. Oh, we'd show them, they'd say, oh, this is really interesting. Oh, really? Do you think it'll work? No. Well, why? And they'd go on and say, well, you know, you're going to have leakage. If you're going to need tight off, it's really tough to get tight leakage in these systems. The cell's not going to like what you're going to put in. They're going to want to stick it out. And invariably, they'd kind of pat us on the head and say, you know, guys, biology is really complicated. You should stick with engineering. Well, what we were able to do at the time is the Office of Naval Research had a pioneering program on gene networks that Eric Eisenstadt ran. And I hounded Eric for about 6 to 9 months, calling him every few weeks, telling him that we really needed money to try to do this experimentally. And we convinced him to give us $500,000 in, I think it was 98, 99, to build this thing. The ONR, like DARPA, likes to have an annual PI meeting where they invite the PIs down to present the work to the other PIs in the program. My wife and I were about to have our first child, so I couldn't go, and I sent Tim Gardner, my PhD student, down. So he basically presented up to here and turned to the audience, and they tore him to shreds. There was a Nobel Prize winner. Everybody basically saying, this is absurd, it's not going to work. And Eric Eisenstadt called me the next day and says, Jim, you're going to have to buff up Tim. He really got attacked. Nobody in the audience thinks it's going to work. I think it's going to work. I just don't think you two clowns are the two guys to get it to work. We now realize you really don't know molecular biology. We're giving you $500,000. I want that money back unless you find somebody who knows molecular biology who can train you and get you guys to work. So he says, okay, don't take the money yet. And we turned to Charles Cantor, and we went to him and says, Charles, what do you think? And he says, crazy enough idea that I think it can work. 2, I think you two are so naive about how hard it can be. I bet you two are the two guys that can get it to work. And with that ringing endorsement, Charles opened up his lab to us, and Tim got in there and actually got trained by a number of Charles's folks and learned enough molecular cell biology to actually get in there. And just very quickly, he began to build different plasmids that he wanted to get into E. coli based on the toggle. So we looked at LAC as well as CI and TET as our respective repressors. So let's focus right now just on the CI-LAC toggle. Okay, so you've got P-trick driving CI, so the LAC wants to bind to P-trick, shutting it down. You got GFP reporting on CI. And then we'll use PL promoter driving LAC, so the CI land repressor wants to bind to PL, shutting it down. Okay, you can, as you many in the audience know, you can induce LAC with IPTG, and we use a temperature-sensitive CI, so you could basically induce that with heat shock. Tim built a number of different versions of this guy, put them into E. coli, and now here's experimental data on the system. So showing 4 different versions. He starts them, let's say, in the off state. Okay, so in the off state, you've got basically CI off, GFP off, LAC on. Okay, now what you'll do is he delivers IPTG, in this case for 6 hours, flips the systems from the off state to the on state, removes IPTG, and there's a lot of variability. It's a pretty big topic right now in synthetic bio and systems biology, and generally gene expression variability. But even though you have variability, these guys will stay in the on state. And our base bistable systems will stay effectively stably in the on state. We've looked out for 96 hours, which are many, 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 many generations for E. coli. But now given them in the on state, you can then hit them with heat shock and flip them from the on state to the off state, and they'll now stay in the off state, again, for as long as you like to look. From an engineering standpoint, if you want to think about using these switches as the front end of sensors, and we'll come to that in a few minutes, what you want in many cases is a really sharp switching threshold. So you want a very clear division between not detecting something and detecting something. And what you can do with bistable networks and nonlinear networks is actually get a very sharp switching threshold. And in this case, Tim was able to characterize it for his system. So what he's showing here is a switching threshold as a function of the amount of IPTG. So you could think of, in this case, using it to sense the amount of IPTG in the environment. So at very low concentration systems in the off state, and then at this point, this concentration, it flips quite sharply from the off state to the on state. And what we're showing here are flow cytometry data at points 2, 3, and 4. So at point 2, you're all in the off state. At point 3, you have some in the off state and some in the on state. And at point 4, you flip them all into the on state. From a kind of device characterization, you also want to look at the dynamics, temporal dynamics, which include the switching dynamics. So in this case, Tim was able to look to see how long does it take for the system to switch. These are all on high copy plasmids, so about 80 to 100 copies in each of the cells. So from switching from the off to the on, it took about 6 hours for full induction, which is captured here as well as shown up here in flow cytometry data, whereas it only took about 20, 25 minutes for the off state. So here we think we basically were looking at cell division dilution effects of the lac-bound IPG or IPG-bound lac, whereas here we just had to worry about essentially having the lambda repressor basically destabilize in the face of heat shock, fall off the promoter, and allow then the machinery of transcription and translation to come on. So what do we do? Well, this is a nice ad that came out around the time that we published this a few months after from Accenture, which was an ad announcing their spin-out from Anderson Consulting. And in this case, what they show was basically fluorescing bacteria, and then this little thing popped up. Bacteria is tested as digital circuits. Now it gets interesting. And that's really effectively what we did. We took motivation from electrical engineering, effectively looking at digital circuit, the classic digital circuit, a toggle switch, a simple form of memory, and created the biological analog to endow cells with memory. As a result of this, there was a lot of interest that biocomputing had been opened up as a possibility. So if you read Scientific American or Discover or New York Times, you'll hear a lot of talk about, in the context of synthetic biology, oh, wouldn't it be neat if you could program bacteria or other microbes that could then be used and harnessed to create computers. I don't think anytime soon that's going to happen. Are you going to have a jar of E. coli or a PC with E. coli inside tagged on to calculate different schemes, in part because of the timing issue, right? The first time I ever presented this was at a DARPA workshop on amorphous computing, where I went through and made the slides that I showed you. And as I get through, the audience is getting more and more excited. Yay, biocomputing is here. The BU guys, woo, have done it. I said, okay, wait a second, guys. I showed that last plot. I said, it takes 6 hours for this thing to flip. All the air went out of the room. Why? Well, did a little electronic switches inside your computers take tiny, tiny fractions of seconds to flip. Imagine now having a computer with an E. coli switch instead of E. coli driving. Imagine surfing the web, see what the weather's going to be. Well, 4 days later, when your website is downloaded, you say, oh, I didn't realize it was supposed to snow 2 days ago. So that's not going to happen. Biocomputing, from that standpoint, I think, is not going to be happening in the lifetimes of us in this room for some of the reasons I mentioned and others. But if you think about this from the notion of programming cells, from a biocomputing standpoint, to do novel functions, we're there. And synthetic biology has opened up a whole host of possibilities. And we got excited on the heels of this with Hideki Kobayashi and Mads Kern and Tim Gardner and others in our lab of wouldn't it be neat if we could use these engineered gene circuits as interfaces, as components to introduce into cells, to interface them with natural signaling pathways or other input schemes, as well as interesting output pathways. On the understanding these things have evolved for millions, if not billions of years, to do really interesting things in the environment. And as many of you can appreciate, there's increasing interest in biotechnology for harnessing microbes for a whole host of functions. So we got excited, for example, could we use the toggle switch, and I'll give you 2 examples on the toggle, as a means to interface to create, for example, whole cell biosensors. So there is, a lot of interest in can you program microbes or other cells to be put on chips, to be put out in the environment to detect the presence of a pathogen, or to detect the presence of heavy metal, or to detect the presence of some nasty agent in the environment. And we made it easy on ourselves. Okay, as a demo, proof of principle, could we use the toggle to make a whole cell-based biosensor that would be a DNA damage sensor? So what we did was we took our toggle, flipped the order, so we used still CI and LAC, but now had GFP reporting on LAC. We replaced the temperature-sensitive CI with the native or endogenous CI, which can be cleaved by REC-A. Okay, so what happens with E. coli is in the face of DNA damage, the SOS pathway gets flipped on, and basically the gateway for SOS is REC-A. So that REC-A gets activated, basically cleave LEX-A, allow the SOS operon and other schemes to come on, flip the response. What we did was set the scheme up so that in the off state, CI is on, GFP, our reporter in this case, is off. So we found that these guys, as I indicated earlier, are incredibly stable. So you want to have these things really stable, right? From a sensor standpoint, you don't want a lot of false positives. Right? So you don't, if you have your cell-based biosensor sensitive to anthrax, you don't want the thing flipping off every couple hours, right? Clearing the, clear building 10, we have the anthrax detection. You want it so that it only flips in the presence of what you ever wanted to detect. Right? So in this case, these guys stay stably off. And what we show is that in the presence of very low amounts of DNA damaging agent, whether mitomycin C, a chemotherapy agent, or UV radiation, you get basically a number of cells beginning to flip on. And in fact, the sensitivity here is better than actually established and characterized DNA damage sensors. We then went further and said, okay, maybe you don't want to have GFP as your readout, which still would be interesting. You could have fluorescing cells in your environment. Could you, for example, have biofilms? Something that's easier to see, so you'd have, say, purple biofilm appear in the field when it detected a mine, or a purple biofilm that could come on if you've got too much UV radiation detected in a lab or out in the environment if you're at the beach. And around the time we were doing this, a group in France had actually identified the TRA gene in E. coli as a gene that flipped on would produce biofilm. So, in short, won't go through these for interest of time, but what we're showing here is our biosensors not in the presence of DNA damage, so no biofilm formed, whereas in the presence of different levels of biofilm, you get a clear biofilm readout. Second key application was one that we got charged with, kind of motivated by, from biotech folks. So Tim Gardner and I talked to a number of different companies when we had come up with some of these SynBio ideas. And the folks in Dow and DuPont in particular challenged us to come up with a synthetic biology solution to basically increase the cost effectiveness of bioreactors. So what happened starting in the 70s or so is that the industry would turn to microbes for producing proteins of interest, whether it's insulin, EPO. And what they'll do is they'll grow bacteria or yeast up to a very high density in a large vat, a bioreactor, and then whack them with inducers to begin to produce the protein of interest. Similarly now, and I'll return just for brief comments in a few slides, the bioenergy industry is doing a similar thing. In each case, you'll grow up a microbe that'll convert, in this case it might be glucose, into, say, ethanol, or it might be sunlight into hydrogen. Okay? In each of the cases, it's incredibly important to be economically efficient, so you want to get your costs down in order to actually make it reasonable to introduce the process. Turns out the inducers are about 10 or 20% of the cost of one of these processes, and it's actually beginning to hold back the industry. They challenge us as, could you program these guys to autonomously flip on protein production when the cells reach a certain density? And so we got excited about this possibility and thought about, well, could we harness quorum sensing and engineer into cell signaling to flip on a gene of interest or protein, have a protein produced when it came up. And we turned to Ron Weiss, who had been an MIT grad student. He's now at Princeton as a faculty member, going back to MIT as a faculty member. And Ron, along with Tom Knight, had basically taken the Vibra Fischeri quorum sensing mechanism. So here's a scheme where basically it's that each cell will produce a small molecule that will diffuse in the environment, and they'll each pick up that small molecule. And you'll have certain promoters that are sensitive to that, so the cells can have a sense of how many cells of the same type are in their area. Ron and Tom had engineered these so they could create sender cells and receiver cells, so the cells could be engineered to talk to each other. In short, what we did was now take this system, couple it to our toggle switch, and use it to flip the toggle switch once you had a critical density of cells. And so you could now produce your protein of interest, and that's exactly what we did here. There's our control, but basically at low densities, no cells are being flipped to basically produce the protein, and then at some critical density, the cells begin to flip until at the higher densities, they're all producing the protein of interest. And we're now back talking to biotech companies and bioenergy companies as to how this general scheme can be harnessed. I want to give two other quick case studies on the synthetic side, and then I'm going to turn to the systems. What I presented here, and really the dominant mode in synthetic biology for the young people out there who are looking at the field thinking about getting into it, really has been primarily transcriptional switches. So that is, can you engineer protein DNA interactions to turn up or turn down gene expression at basically the transcript level? We got excited in our group a few years ago about, could you do this from a post-transcriptional side? So could you take advantage of all the marvelous regulatory roles that have recently been identified for RNA and harness those to engineer interesting switches that could also be utilized for a number of different applications. So with Farron Isaacs and Dan Dwyer, who are grad students in my lab, they basically came up with this notion of engineering riboregulators. In fact, this was motivated by seminal work by Susan Gottsman, who's here, on and kind of inspired by natural riboregulators that exist in bacteria. So what these guys did was say, okay, you've got your general prokaryotic gene expression where you'll have transcription, you'll have your mRNA produced, the ribosome will come in, dock to the ribosome binding site, chug along, translate the protein. Okay? Susan Gottsman has shown that you have in some natural systems that the ribosome binding site will actually be blocked, and you'll then have a small non-coding transactivating RNA that can come in and basically expose the ribosome binding site, allowing it to be expressed, and some really interesting interactions. And so Farron and Dan, using that as motivation, said, okay, could we engineer schemes that could do the same? Could you come in and engineer a cis-repressed sequence that would be designed so that when the mRNA was expressed, the cis-repressed sequence would bind and block the ribosome binding site, preventing the ribosome from coming on and producing protein. Okay? That's step one. So you could have this shut off. Then as step two, could you now then express when needed a short transactivating non-coding RNA that would be designed specifically to bind to the cis-repressed sequence so that you had some probability of it binding, pulling it off the ribosome binding site, exposing the ribosome binding site, and now allowing the ribosome to come in, chug along on the mRNA and produce the protein. What the guys did was they did a number of different bioinformatic analyses using MSOFT to design a number of different cis-repressed sequences as well as transactivating sequences with different levels of complementarity. And so here's an example of a cis-repressed sequence where in blue is a ribosome binding site. They then designed corresponding transactivating RNAs, shown here, and actually used the models to predict how well they would interact. In this case, the prediction was that, in fact, it would expose the ribosome binding site. And then Farron and Dan, on the basis of a number of these predictions, got them designed and into E. coli. And what we're showing here is, here's our background negative control. Here's basically a system without the cis-repressed sequence. When Farron and Dan introduce the cis-repressed sequence, they can get 96 to 98% knockdown, which is a huge amount, very high level. And then when they express this transactivating scheme, they can actually get 10 to 20 times boost up of an expression above that very tight knockdown. What they found is you can have a very fast response. You can begin to detect protein about 5 minutes after you express your transactivating RNA, right? You can appreciate that because you basically have a whole boatload of cis-repressed mRNA sitting in your cell, and now all you need to do is transcribe a very short transactivating RNA to get the now translation machinery going. As I said, you can tune it, so it's a rheostat. You can go from effective off, tune it up to different levels of expression. And of big interest to us, you can get high specificity. So you can basically design a bunch of different switches that can all act in cells together, so you can create a switchboard, which is one area that we're now working on. Could you have a switchboard to tune up or down the expression of multiple genes independently in a cell? And one of the key motivations in that is, again, going back to the bioenergy space, is there's real need to increase the efficiencies of basically how you shift carbon in the cell to your pathway of interest and away from some other pathways without building up a lot of biomass or toxic problems. As you can also appreciate, this is highly modular, so you can put any gene of interest under its control. And I'm going to give one case study, SynBio, and then link it to CysBio in our first application there. Key application on this was after we did this, and as synthetic biology began to get a lot of attention, not so much for what we were doing, but from some high priests who were talking a lot to the media, there were, as you can appreciate here at the NIH, where I know there's a good component on ethics and discussion, a lot of concern that the engineers were running amok in these molecular biology labs. And increasingly, kind of harking back to the early days in genetic engineering, there were growing issues of concern. Would we create Franken-microbes that would escape from our labs and do nasty things? So would Collins' lab do something where the microbe would escape, eat City Hall, or worse, eat Fenway Park and wreak havoc in the city? Okay? As for many reasons that we can discuss, I'm happy to. We're really not anywhere close there. We're doing very baby steps. It's really essentially genetic engineering on steroids, but really weak steroids. Anyways, with this in mind, we got intrigued on the possibility, well, wouldn't it be neat as engineers to directly address the problem by, for example, engineering a synthetic gene network that can count? So could you design a network that could count, for example, cell divisions and or light-dark cycles so that if you did put something out in the environment as a biosensor or in a reactor or whatever scheme, that after some period of time it would self-destruct. So you'd program it so that it would actually express a toxin so that it would kill itself after 10 cell divisions or after, say, 5 days. So with Ari Friedland and Tim Liu, in a piece we've got coming out in a couple weeks in Science, we came up with two different designs for genetic counter. I'll highlight one, where we basically use the RNA switches of the riboregulators I'd mentioned in a daisy chain cascade. So in short, what we do is we have now set this up to detect pulses of something of interest, which might be a protein produced as part of cell cycle. Here we did it just as detecting arabinose. Okay, so you've got cis-repressed sequences that are designed, as I said, in this daisy chain cascade. So, for example, you'll have this scheme set up so that in the 0 state, your system is naturally producing cis-repressed T7 RNA polymerase. Okay, your system's sitting there waiting to detect a pulse. With your first pulse, what will then happen is that the pulse will drive the production of the transactivating RNA, which will now interact with your pool of cis-repressed T7 RNA polymerase, produce T7 RNA polymerase, which will now trigger your downstream promoter, which will now produce a pool of, in this case, cis-repressed GFP. Could be any protein of interest. Okay? That's now sitting there waiting for the next pulse, and you basically, this guy will die out, your transactivating RNA. So then with your next pulse of arabinose or whatever you want to detect, you'll now produce another boatload of transactivating RNA that will now interact with your cis-repressed GFP and produce your protein of interest. We did a bunch of different analyses on this, but actually got this built into experiment and into E. coli. So these are real experimental data from E. coli, where here's our background uninduced. You get some leakage through. So on just one pulse, the first pulse or the second pulse, so these were delivered pulses, I think about 10 minutes in duration, separated by, say, about 40 minutes. You got a little bit of leakage with just the first delivered or just the second delivered, but you got a very large boost, large dynamic range after the 2 pulses were delivered in sequence. What you could do is you can linearly extend this to count up to 3, 4, 5, 6, 7. So we actually did it to 3, where you just tag on other components with different RNA polymerases. And we similarly got really good data. So again, you have a little bit of leakage, which you can tune with protein degradation on your different components. But you get a very large dynamic boost when you actually hit your 3 in a row. We wanted to use modeling to see could we gain insight into how this behaved and could it point us toward optimality. So with Xiao Wang, a postdoc in the lab, he developed a very nice mathematical model based on the 2 counter and the 3 counter. So here are the data in which he basically fit his model in order to get parameters. And then what we see here are predictions that are shown as the closed lines of how would the system behave as a function of the pulse length versus the pulse interval. Right? For the electronic guys in the audience, you have a certain region where, okay, if you hit it too close, it's going to see that as one long pulse and not 2 pulses. If you have a really long 1 pulse, it'll also see that as 2 pulses. But it turns out that we then were able to use these as predictions to say, okay, this looks like the best region. So this is now plotting what's the size of the nth pulse, the output of the system after you hit it with n pulses, and what's the dynamic range. So this region of kind of intervals of 20 to 40 minutes and pulse lengths of, say, 20 to 30 seem to be the really nice region. And what's beautiful about this result is that our circles are the actual experimental data. We went in, tested what the predictions were of the model, and it turned out that our experiments very, very nicely matched what was predicted. Tim Liu then went further. I actually won't show you these data and the scheme, but we have a whole different counter that was produced that was based on recombinases. So that now on the detection of the pulses, it would actually flip different, basically, chunks of DNA to flip on in a daisy chain cascade again, downstream components. And whereas this guy operates on the order of minutes, 10 to 40 minutes, that guy operated on the order of hours, 10 to 20 hours. So whereas the former you could couple to the cell cycle, the latter you could have alluded earlier, use it to count basically light day cycles. So what can you do with these? We got excited. When Tim and I first got into this business in 99, 2000, we were very excited about the notion of using these in the context of cell therapy and gene therapy. Right? And that field kind of imploded on itself. It seems to be coming back, which is nice. But as many of you can appreciate here at the NIH and the areas, is that the challenges there are how can you get safe vectors in a gene therapy case, and how can you get effective encapsulation technologies for cell therapy, where you take cells out of your body, transfact them with construct of interest, put them back in your body. When we get there, the big challenge will be how do you regulate them reliably and safely. Right? As all of you can appreciate who do wet work, you already have catalogs from which you can buy genetic switches. Right? But all of those switches are pressure type switches. And that is, it's as if you come in the room here and turn the lights on, you have to have your finger on the light switch to keep the light on, or have your finger on the light switch to keep it off. That is, you always have to have a chemical present. And the toggle switch, it's like the lights here. You only have to flip it on transiently to keep it on, or flip it off transiently to keep it off. And that is, the chemical, you only have to say take the drug once to basically flip on your construct of interest, and say flip it, take the pill once to flip it off. When Tim and I went to talk to drug companies about the idea, they kind of looked at us in disbelief. And we says, what's the matter with you guys? And they says, what's the matter with us? What's the matter with you? They says, what's your business model? I says, well, it's good for compliance. You don't have to take drugs all the time. Side effects. And basically they shook their head, just like this guy. In the audience where it says, do you understand what we do? I says, what? They said, we sell pills. And I says, oh, well, you could just make a really expensive one pill. But basically they would call us a taxi and usher us out. The idea there, though, and again, you can appreciate this from the safety standpoint, is that I think in the end, things like the toggle switches will become quite valuable, really from a safety standpoint. That, particularly from gene therapy, when you get these things into your body, it's going to be very difficult to get them out. And in many cases, it's going to be difficult in our lifetimes to predict, in many cases, how somebody's going to respond to all the different things you could put on. And so it would be a shame to have somebody take drugs to keep off a gene or protein that was put in to help them out, and as a result of bad reaction. So it'd be nice in this case with the toggle, it could be an FDA safety switch. You could take it to keep it off if you had the bad reaction. Quickly, I already mentioned biosensors and bioenergy. The bioenergy is an area that's really getting a lot of attention from the synthetic biology community. But really less Flash Gordon and more short term is we got excited from a functional genomics standpoint. And that is, could we use our synthetic biology approaches to begin to gain insight into how genes function and how cells respond to different conditions? And more critically, could we use it to study natural networks? So we went back, this is now about 2002, with Tim Gardner and Stephen Young and Jesper Tegner in my lab. We got excited about the idea of, wouldn't it be neat to use our synthetic circuits as basically a little toolbox to go in to probe cells, measure the response, and from that infer natural circuits and natural networks. Kind of on the heels of the genome projects, with these large parts lists coming out, the big challenge for us in biology broadly will be understanding how they function. And these things function in cell networks, in pathways, and circuits, and it's directly linked to their biology. So we got excited, well, why don't you go in and probe it? And really it was motivated from, again, electrical engineering analogs. So again, any electrical engineering student in the audience will know that as an undergrad, you'll invariably have a class where the professor will give you a circuit board and say, okay, hotshot, how is this wired up? And they don't give you the wiring diagram. So what do? You run into the electronics lab, you turn as many of the components as you can in sequence on or off or up or down, measure as many of them on the board as you can, and from that begin to infer how they're wired together. Okay? Most of you are not electrical engineers, but many have bought a new home or been involved in renting a home where you might not have the wiring diagram for your house, right? But you have a circuit breaker box in the basement. So what do? Take your boom box, turn the volume up, plug it in the kitchen, you run downstairs, you flip the switches, right, till you hear the volume go up, kitchen, right? You run back upstairs, plug it in the dining room, run downstairs, flip the switch, dining room, okay? And you do that, you get a first order map of your house. Okay? Then you get your second order map by trying to microwave popcorn in your kitchen while you have the air conditioner going in your bedroom. Poof, you blow the circuit, okay, there's a nonlinear interaction between those two, and you make a point, which you never seem to pay attention, and you do that repeatedly. But you basically, by probing your system, get a sense of how they're wired together, and that's effectively what we did. We did a whole series of computational studies, then experimental studies in E. coli, in yeast, in mammalian systems that allowed us to reconstruct these networks. We then got excited as engineers to think about what could you do with those networks. And I'm going to talk about a number of different aspects, but the one that got us most excited was the idea that we realized you could use it to identify the mode of action of drugs. Okay, so what we do on our analyses is the output is a quantitative network model that tells you which gene is talking to which gene, what's the nature of the interaction, is it excitatory or inhibitory, and what's the strength of the connection. Okay, so you get this quantitative network model. And we realized, hey, you could actually use this to identify what's a drug hitting. And the value for that is, again, for those of you who interact with biotech and pharma, is that in the target-rich post-genomic period that we're in, companies are really good at setting up assays to see, does the compound under development hit the target that it was designed to hit? Okay? What they can't do, in the most part, is tell you what else does it hit. And it's these other hits that are posing real challenges for side effects, as why a lot of drugs fail in phase 1, phase 2, phase 3, or pose problems when they're actually approved and out there, because we don't know what they're hitting. Well, we realized, hey, you could actually use the network as a way to gain insight. And here's the way we thought about it, is that I teach control theory to our undergraduates at BU. We just finished the semester. And from lecture 1 to lecture 28, what I tell them is you have 3 parts to a system: the input, the output, and the system itself. If you know 2 of the 3, you can work out the 3rd. Okay, so in fact, the way we work out our network model is we go in and make a whole series of inputs, measure the outputs, and from enough coverage and measurements, we can actually figure this out. Okay, so now imagine you've got a drug that you don't know what it's hitting, but you can measure the output with some expression profile, let's just say an array. Okay, so you have the output. Now, if you have a good model of the system, you've got 2 of the 3. You don't know what the input is, but if you take your output, you can filter it through your network to predict what was the likely input that actually produced this output. And what we're able to do, firstly in silico, and then in a series of studies in E. coli, yeast, and mammalian cells, was to show, in fact, that that could actually work. What we then got excited about was different applications, and what I want to do is just highlight a few in the bacterial space, and that is we got very excited about using these approaches to gain insight into how bacteria respond to antibiotics. Why? Well, these plots are very familiar to many in this room, is that the number of resistant strains, both in hospitals and the community, are going way up, and yet the number of new antibiotics and effective antibiotics being produced by pharma is going way down. Right? Pharma is in many cases closing R&D efforts in antibiotics. It doesn't monetarily make sense to them. Right? They haven't figured out how to sell bacteria Viagra. If they could, we'd have vibrant antibiotic efforts from many of the major pharma. Right? So you've got this nasty contrast in increasing and decreasing, and we got excited. Could we use our approaches to begin to gain insight into what are the antibiotics actually doing? And our starting point was looking at reconstructed networks. And so this is work done by Jeremiah Faith and Boris Haidt in my group and Tim Gardner's group, where we've actually collected hundreds of E. coli expression profiles that are available on a many microbes database. It's publicly available. You're all free to play around with it. And we had developed mutual information-based approaches that allowed us to reconstruct these networks. We then used this genome-wide basis in E. coli to begin to explore how do actually drugs act. And our interest was actually looking at quinolones to start off with. So we looked at norfloxacin. There's a gyrase inhibitor. So quinolones act by basically inhibiting gyrase, damaging DNA, and it's thought through that action that will basically mess up DNA replication to be the primary way in which they kill. We wanted to see, could we use our network-based approach to gain insight into other ways in which these things are killing. Okay? In addition, kind of it is an integration step with synthetic biology, is we used the riboregulators that we engineered to also control CCDB, a toxin which had been difficult to study before because it expressed at low levels and the type of expression vectors you typically would have, cells just would either die or they wouldn't grow. We used both these in combination with our network approach to see, okay, what was being induced, what were the key mediators? So it's not simply looking at differential expression. We're actually using the analysis, the network base, to try to figure out what actually are the mediating factors, pathways, or networks. And what came up were some that weren't surprising, DNA damage response, but others, oxidative damage response came up, iron uptake and utilization came up, as well as the iron-sulfur cluster synthesis networks came up. Dan and Mike Kohansky went in, did a series of knockouts and phenotypic studies in overexpression to basically validate a number of these findings and arrive at a mechanistic model that basically indicated that both quinolones and CCDB, in addition to their primary actions, also induce an oxidative damage pathway that leads to production of hydroxyradicals. And in short, basically what they found is that as a result of the action of the gyrase inhibition, superoxides generated that will basically interact with iron-sulfur clusters, leading to the leaching of iron that will feed the Fenton reaction to then produce highly lethal, damaging hydroxyradicals, which, as you all know, can damage DNA, lipids, and proteins. Mike Kohansky, who's an MD PhD student in my lab, brilliant guy, went further and he says, okay, if in fact you've seen this with quinolones, why not explore to see if in fact this is coming up with all classes of bactericidal antibiotics? Oh, really? Okay, so with cytal antibiotics, what you have is basically 3 different classes. You have your quinolones that damage DNA, aminoglycosides that mess up protein synthesis, and you have beta-lactams that mess up cell wall synthesis. Each thought to act differently, leading to death. What Mike speculated was that in addition to their specific targets and actions, that perhaps these guys are also stimulating the oxidative damage pathway, leading to the production of hydroxyradicals. And in the interest of time, I'm just going to basically say that's in fact what he found. These guys, in fact, did produce hydroxyradicals. When he looked at the statics, so bacteriostatics are now antibiotics that don't lead to death. They actually lead to growth inhibition. None of these were producing hydroxyradicals. Mike then went further. What's the implication of this? The implication of this, say, common pathway, and he expanded it to find that there were also metabolic interactions with these guys, basically mess up the TCA cycle, that mess up the NAD, NADH ratio, that then produces superoxide, feeding the Fenton reaction, et cetera, is that if you, in fact, have each of these producing hydroxyradicals damaging DNA, then what you have is that if you could block the DNA damage response, in principle, you should be able to potentiate all cytal antibiotics. And which he shows this genetically. He knocks out REC-A, and he finds that it can even get multiple orders of magnitude improvement of killing with these antibiotics. So the idea there is that as a result of this common mechanism, you could come up with a small molecule that could lead to super Cipro or super ampicillin or super gentamicin. And what we found with some screening work that we did with Guillaume Cotterell is in using a REC-A ATPase assay and looking at a number of different, actually NIH-supported libraries, one at BU and one based at Harvard. We looked at a number of different compounds, found a number of different hits that, in fact, in this one, CB301, here's a compound that by itself does absolutely nothing, but when in combination with gentamicin, actually improves the effectiveness of gentamicin by 3 orders of magnitude. And so we're now working with Scott Singleton at UNC, and we're initiating a collaboration with Stu Schreiber, where we're going to do additional screening and medicinal chemistry work around these compounds to see can we get to a small molecule. So we're down to just a few minutes, so I've got at least one case study, possibly two, but I'll quickly go through it. Kohansky went further and said, okay, as all of you can appreciate, radicals are mutagenic, and as a result, this would imply that the antibiotics themselves are mutagenic. Okay, so that delivering cytal antibiotics would themselves produce mutations that could lead to resistance, right? We generally think of resistance as arising either as a result of the random introduction of mutation that then is selected for, or by the sharing of plasmids that have resistant genes. Kohansky went further and says, well, maybe they themselves are mutagenic. And what he was able to show was, in fact, that there was a significant boost up in the mutation rate with the radicals, with the delivery of the antibiotics, and it's directly correlated with the radicals. And that when you either deliver thiourea, which is the radical quencher, or you consider this in anaerobic conditions, the effect disappears. Okay? So now you go further and say, okay, what does this imply? Well, he was then able to show that if you now deliver sublethal levels of antibiotic, after a number of days, you're going to have resistance emerge to a large number of antibiotics, including those that aren't actually the ones that were applied. Okay, so in short, what we're showing here, after just 5 days of sublethal levels of ampicillin, that not only do you get resistance developed to ampicillin, but you also get resistance to a range of antibiotics, for example, norfloxacin, as a result of the mutagenic properties induced by the antibiotics, as a result specifically of the hydroxyl radicals. So when you actually deliver on anaerobic conditions, the effect has effectively disappeared, which now poses and introduces some pretty scary notions about the overuse of antibiotics, whether in our foodstuffs, around our homes, or if you're taking lower amounts. So clinically, low levels of antibiotics can lead to these low levels of resistance. So even further is when Mike actually looked at this, for example, looked at the quinolones, what he found was that there were several strains that were produced by ampicillin, but not resistant to ampicillin. Okay, so as a result of kind of this non-selective introduction of the mutations, it can now lead to some pretty scary situations. Okay, so I actually was given a 3-minute warning, so I actually have time to just very quickly give a very exciting study that another student in my lab did, which is Carrie Lauren. So Carrie's a PhD student, went back into our arrays to look to see were there other things being induced by the antibiotics. And in short, what she found were a number of genes that are NO sensitive, nitric oxide sensitive. Quickly just going through this, she then went and used a number of different schemes to see was nitric oxide being produced by different cytal antibiotics, and in fact found that was the case. That is that the antibiotics, in addition to the radicals, were basically inducing the production of nitric oxide inside the bacterial cells. When you actually knocked out NARL, transcription factor involved in, it can be involved in NO production, particularly under anaerobic conditions, you basically had a significant reduction in the amount of NO being detected, and you actually had a protective effect. So that is, it appeared that the NO being intracellularly produced or induced by the antibiotic was actually contributing to the killing and making them more effective. So it was basically interacting with the other effects of the antibiotic. Carrie went further and said, okay, I wonder if in fact NO is a small molecule, signaling molecule, well studied. Was the NO being induced, the NO being produced as a result of the antibiotic induction, being signaled to other antibiotic cells, any other bacterial cells in the population? And in fact, that's what she detected. She could actually find they were signaling, and actually discovered that those that received the NO as a result of the signaling before the delivery of the antibiotic actually had a protective effect. That is, that they were protected by, basically they could delay the killing on the order of a couple hours. And so Carrie was able to arrive at this idea that in addition to the radicals being produced, that the bactericidal antibiotics are also, as a result, didn't go into detail, but messing up respiratory chain, particularly anaerobic enzymes thought to be involved in anaerobic respiration, leading to the production of NO that messed up a number of different proteins, including FER and FER-regulated proteins, that contributed to cell death. So you had basically this kind of binary mode, that the intracellular effect was cytotoxic, whereas the NO produced would also be signaling to other cells that could be protective. Short, where are we going with this? We're taking our network biology approach to look at persisters. So these senescent cells in any in a population, a very small number that you can't kill. We're using our network approach to try to understand how they're formed and how can you kill them. We're looking into now pathogens, including TB. We're also, again, inspired by work from Susan Gossman, looking at small RNAs. Can we use our network approach to begin to identify small RNA regulatory networks, as well as potentially identify novel small RNAs? We are looking at other higher organisms. We're now using our approaches to look at mammalian diseases from a number of different standpoints. But in interest of time, I want to just finish with this great slide. Born to Network is networking in your genes. Well, it wasn't for me when I was first received support from the NIH, but as a result of some really good, fun developments at BU and great support, and now marvelous support from the NIH. I want to gratefully acknowledge the Pioneer Award through the NIH Director's Office, which allows us to explore all these really fun, clinically relevant areas, particularly in antibiotics. And I encourage each of you, this is our lab website, we have all of our papers of relevance up there. And so with that, when I open up for any questions.
We do have time for a couple of questions.
Okay.
And at 4 o'clock, we're going to head out for the reception.
Okay.
I do have a question for you about the synthetic biology.
Yeah.
People get to the microphones.
Okay.
How frequently do you end up with off-target interactions? So you design intended interactions between proteins, but the host cell is certainly there. How frequently do you run into unexpected interactions?
Yeah, so the question was, how often do we have to deal with unintended interactions from the synthetic biology standpoint? That is, you have your system, you assume it's isolated from the host. In what case do you see interactions? We haven't been bedeviled so much by that from what we could see. We try to pick proteins which generally aren't interacting with much of the rest of the genome. The challenge, and I mentioned this at the lunch with the postdocs, that we have found, and much of the field has found, is that it's very easy to come up with your little schematic. It's very easy to come up with your model. It's frankly pretty easy to now put it as a plasmid or create your construct. It's easy to get in the cell, and it's easy to look at it. And it's really hard to get these things to behave in the way you want. So that first step might take you a few weeks. To actually get it to behave according to spec can take months, if not a year or years. And it's a big challenge in synthetic biology. I mean, there's a lot of talk about, from a bottom-up approach, taking components, putting them together. But frankly, we don't have these components particularly well characterized. We often don't know their interactions well enough to actually do the modeling or those measurements well to have the predictability that the electrical engineers have. And so that's where the challenge is. And I said to the postdocs at lunch, my lab has actually shifted much of our efforts in synthetic biology away from designing to functional spec to instead designing it toward architectural spec. So we'll make the network, and now instead of trying to get it into this little parameter space, we instead just characterize its dynamics. So now the potential bug becomes a feature. So you can say, this thing behaves this way, isn't that nice? There you go, blah, blah. As opposed to, man, it's going to take us 3 years to get a mammalian toggle switch going.
Hi, I have a question regarding the sensors.
Yeah.
So when you design the sensors, you have to predict the operating temperature or what is the condition that the sensor is going to work properly, right?
Yeah.
So can we do that with biological systems, because it's much more complex, but when you go for electrical engineering, you have the device characteristics to begin with. But in this case, we have all set of interactions, right? So how do you work around that problem? So
it's an interesting question, right? So you're getting after sensitivity really to environmental conditions. And many microbes have actually evolved to be really quite robust under a decent broad range, right? So I suspect, and yet you have things like transcription, which is going to change depending on temperature. So can you adjust for your different schemes as you make adjustments? And I suspect what you probably would want is to couple it with a relatively smart electronic component, right? Take advantage of the specificity you can get with biology to detect your element of interest, but have it coupled to a chip via biofilm or in a spore-type arrangement that could account for other environmental factors, whether it's the level of oxygen, the level of light, the level of temperature, pH in the environment, to say, okay, this is actually a true detection event, and you have to account for that the expression, for example, gene expression changed.
And one more question regarding control systems. So if you have input and output, you can predict the behavior of the system, but how much detail you can get from the system? I mean, like, this is the input, this is the output. You can design the system using differential equation or anything, but can we say that, okay, this part of the system is composed of this, or this. So how much detail you can get from the input-output characteristics of the system?
Yeah, another really good question. So certainly many in this room have for decades done very detailed studies that can actually feed the types of models that we do either as predicates, priors, to give you additional info. Really the trick that we've tried to do is on the basis of relatively small, a relatively coarse level of measurement, albeit done at a high coverage, that is, say, for example, microarray data. Can you actually infer decent black box arrays, networks on the basis of the arrays? You know, it's a challenge. It's more so, and I guess your question really gets at a fundamental challenge is that it's how do you get after kind of the singular network, given that in principle there's really an infinite number of possibilities that could account for what you see. And there's a number of different tricks. I mean, we basically try to get after the best fit, as most of us in the room would. But things, for example, like sparsity, you can actually exploit. So many networks are not highly connected. Most biological networks are highly sparse. So that is, any given gene, it has very low probability of being connected to any other gene. And it turns out, strangely, that from our analyses, the most sparse network predicted by the approach tends to be the correct one.
When you have switchable networks, the effects of noise may be significant if you're going for in a binary switch. Have you done analyses of the effects of noise on some of these networks? And is there some way to actually use the response of the networks to learn something about the underlying fluctuations in the system?
Yeah, so it's another great question. Yeah, we had and we still have a very active effort in our lab around noise. In fact, that was part of our way to get in from the modeling perspective from our efforts of introducing noise into the body to improve balance control, where one of our first pieces was actually designing a noise-based switch that both was a switch and an amplifier. And so we studied experimentally what's the role of the fluctuations to flip you from one state to the other. Carson Chow, who's here, as I mentioned, his group is actually looking at that now from a stem cell differentiation perspective. Others are actually trying to use the noise as a means for system identification. So it goes back to the prior questioner. In engineering, you commonly, instead of, say, putting in an impulse or putting in a periodic wave, you'll put in noise as a means to then pull out the underlying structure. And Mike Simpson at Oak Ridge National Labs and others have now introduced noise as a way to begin to probe the underlying structure on the scheme. You know, the challenge, and I was talking with Susan Gottsman earlier, is how do the cells deal with all this noise? It's amazing. When you actually dig in and look at it, these things have high levels of fluctuations, and yet, for the most part, they're pretty happy. They're doing their thing. And much of the area in synthetic biology in this space has primarily, up to now, focused on characterizing the amount of noise and beginning now to study the phenotypic consequences of those noise. But we really haven't gotten a good handle on what are the ways that the cell can actually regulate, control, maybe exploit the noise when it wants it for differentiation, but say lower it at, say, low expression
That's
levels.
what I was going to ask in some of these control systems and applications, let's say in bioreactors, whether you can use the response of the system in some way to probe the fluctuations in the environment.
Yeah, so it's a great point, right? So the easiest way, right, is to make your threshold above the fluctuations, right? So if you can do your analysis and say, okay, the probability now that this guy would ever go above this threshold is none, is zero. And so if we had this up here, at that level, you'll never flip it. However, you might also want to exploit it for an oscillator or a very sharp switch where you would be on this region and have a secondary level of control. But we've actually been talking to these guys about how you can tune the threshold on these switches. Coupled also to that genetic counter that I showed, in order to deal with leakage, we're actually now coupling it to the toggle switch so that you won't flip the output until you actually exceed some level of scheme, so that now it's basically, you have no leakage through your system.
Thanks.
I'm afraid we should probably stop there and ask you to hold your question for the reception, which follows immediately. Please join me in thanking our speaker today for a terrific talk.
Thanks, Eric.